All right, I think I'm going to start introducing our session to make sure that we have enough time to cover all the great content we have lined up. So we've had a really full afternoon so far. We started just after lunch talking about the state of Medicon Valley. We had our new CEO, Annette Steenberg, go through some facts and figures, life science in Denmark, Sweden, as well as the beacons of Medicon Valley. Once again, if you're interested to look deeper into the numbers, there are hopefully still some hard copy left at the back. Otherwise, go online to our website and you can find more information there. After that, we had a discussion on framework conditions for life science and innovation, talking about the value of collaborating much more across Denmark and Sweden from a larger perspective. We then looked at funding and investments and how a startup should navigate in that space, relate to US investors, and so on. And it's really great to know that Jantelov may not be such a bad thing after all, something we should really be proud of. But now we're going into, innovation in the context of clinical research and private-public partnerships. And once again, 3 really great speakers talking to that theme. And just like the previous session, we will have the 3 presentations first, and then we'll take all the questions from you after the 3 presentations. So make sure you go into our mobile platform. For those of you who are not aware, you can scan the QR code behind your name tag. You access our program, the Q&A part, and please let us know what you think of the different presentations. So to start us off, it's my great pleasure to invite our first speaker for this session, Stefan Yerwinga, who was recently appointed Research Director for the Skåne University Hospital. Stefan is also a professor at Michigan State University, University of Texas, and Stanford University. So a fantastic international and Swedish connection. So Stefan, you're very welcome on stage to walk us through your presentation. The floor is yours.
Thank you.
It's the clicker.
And I want to thank you, the organizers, for inviting me to talk about large data and the opportunity for the future. And my background is I'm an intensive care cardiologist interested in both basic science but also clinical science. And I served as a research director in Michigan for almost a decade. So I apologize already now for presenting data out of US as I don't know fully. I just arrived to Sweden. But I try to progress. Okay, so yeah, thanks. So first, disclosure from my universities and also that I'm an advisor for a single-cell technology company. So, as we've seen many times before, we face a challenge which is quite depressing, that we getting older and by that consuming more health. Even if some try to present data that the healthy 80-year-olds or octogenarians are more prevalent now than before, but still the burden on our healthcare systems do increase. And our main way to address health issues is getting more and more complicated. So we know already now that for every 9th year, we halfen the number of drugs we get out $1 billion from ideation to FDA approval. And actually, we don't even get one drug. So right now, it's about $3 billion to get something from ideation to FDA approval. Here comes the first set of US data. So these data have mainly been generated through having, at the time of the event of the studies, and based on collecting information at the event and before planning the studies. However, we have an enormous opportunity in getting more digitalized. So it's even more pronounced in Scandinavia, and actually the landscape is even darker today than it was 2013. So a lot of health information is available in different databases. However, it's mainly on sick people. But even here, technology works in favor of us. So if you look on the tech development, so the devices connected to internet is exponentially increasing and predicted to continue exponentially increasing in the future. And what's even better, it seems like we are wearing the devices to a larger extent. So even a watch of today, everyone here having an Apple Watch has an FDA-approved device for ECG surveillance. But what takes the opportunities even further is that the possibilities with wearables is increasing. So there are already technology today to measure pH, even if it's not on the market. And actually, most common lab chemistry can be done through wearables today on the experimental level. So don't limit yourself in your imagination to what type of your daily activities can be covered through data sampling. That's one challenge. It's a huge amount of opportunity, but it's a huge amount of data. But it's not only the amount of data, it's also the granularity. So this is the cost of sequencing the whole human genome, which at the beginning of the Hugo project was $100 billion. Now it's approaching $100 per individual. And that gives an enormous opportunity for granularity. The biggest challenge is to stay granular with our data. As we get massively invaded by data, we need to keep it as granular as we can. We tend to summarize our data. So by summary statistics is the most similar, descriptive statistics, but those descriptives can actually represent a lot of different data. So that is the key another key challenge. So, and when it comes to tissues, and as we heard about proteomic and the human protein atlas, we don't talk about tissues as tissues. We talk as a bunch of single cells. We have delineated cardiomyocyte generation in adults by being able to find those few cardiomyocytes which are drowned in the data of a human heart. So, so this is the slide to remember after my talk. We're going to have an exponential growth of data. And if we do it well, it's not only by sources, but also by granularity. So taking this to the opportunities. So one is, of course, generating predictive models through AI, but one is to be able to address rare diseases, which do generally not present a lot of data. Big Pharma has no solution for these individuals because in US, the definition is that less than 200,000 individuals are afflicted by the disease. And that does, of course, not represent the market for to return of investment for your $3 billion. So these patients are left without any specific treatment. Are these not important for public health? Well, in US, about 10% of the population has rare disease. So in US, only more than 30 million people are inflicted. So what we did was, in my research group, was actually to start to look at all the investments we had done in the drugs, the $3 billion paid for just one thing, the one-trick pony model of drugs, and actually view drugs as three-dimensional structures, which do not only have the epitope which we define its interaction by, but has other epitopes. First thing was to go into my own area, heart failure, and look, do we have any indication in literature that this could be true? And the pivotal drug in the field of heart failure has been to inhibit a protein called angiotensin II. And we know that if you do that by inhibiting the rate-limiting enzyme, we do reduce mortality significantly in these patients. And as you see, if you treat them, and please disregard the legend which is switched around in the original publication. It's actually the lower part of the figure is when you do inhibit the rate-limiting enzyme. You see that you actually get the reduction of the protein, but after 80 months, you actually have some kind of escape mechanism, so you get it back to the original levels, even if you treat. Do you get any loss in mortality? No, you still have the effect. And if you would have an escape mechanism to generate this, then blocking the receptor would be the solution. And as you see in the second figure, there is no difference if you actually block the receptor or you block the rate-limiting enzyme, indicating that what we have as a fundamental drug principle doesn't work according to our drug principle. But no one wants to withdraw the drug because it's a major mortality reducer. So how would we screen for this kind of effects? As we know, all the effects in cells are generated by gene expression through an RNA template. And the main focus has been to look on the effector molecule, the protein. But the problem with the proteins are that they are modified. They are generated some instances as pro-inactive enzymes, and when they're active, they're actually being inactivated by the cells. But all these forms have an imputation. They impute on the gene expression. So if you know the gene expression, you can delineate activities of these proteins. So then, how would we high throughput screen for these effects in this rare disease by using massive data? Well, if you take each drug and treat a cell population with a drug, and you look on the differential gene expression caused from the control cells, you will actually get a fingerprint of that drug's activity. And now, if you go back and look on drugs having the same drug target, we actually see that they have similar but not identical patterns, proving our hypothesis true, that there are unique effects which are not mediated through your ligand definition. If you do it on 17 different cell lines, or representing 70 different cells and tissues, and you do it with 20,000 compounds, you will generate 1.3 billion data points. And once again, keep it granular so you can go back and study the details. And we studied a cardiomyopathy, which is a prevalence of 1 in a million. And these patients do get an early, very arrhythmogenic cardiomyopathy, and there is no treatment. They get a traditional heart failure treatment, won't help them. They need a heart transplant by 40. So what we did was similar to the drugs. We actually did the gene profile of the diseased cardiomyocytes and the healthy cardiomyocytes. And then you could get the differential, the pattern of the disease on the gene expression level. And then we could match to our drug database, where we had profiled each drug for its imputation on the gene expression. So what drug did best reverse the disease pattern? And by doing that, you do enormous statistical testing. So there is a mass significance for false discoveries. But by coupling this to testing on the cardiomyocytes, we could actually show that we could correct the arrhythmia. By one of these predictions, we could actually correct the lack of contractility and the lack of relaxation only in the diseased cells and only with one of the predicted drugs. And we can actually correct the pathways at the protein levels. So this is just one example where you could use massive data even in rare diseases where there are not many patients. So I just wanted to bring that as an example, because generally when we talk of massive data, we talk of large disease groups and patient flows and large patient flows. But large data gives us an opportunity to address rare diseases as well. So this is the flow that you actually characterize all the drugs by the gene expression. You take a blood sample from a healthy individual, a blood sample from a diseased individual. You do reprogram cells to actually get diseased and healthy cardiomyocytes. And instead of $3 billion, you enter the market with less than $50,000. Actually, I would say even half that. So this is just an example of how we shouldn't be too depressed about the curves. We see all the time that older population increases the need of healthcare. The cost of drug development has gone over the edge, but we have opportunities with big data and how well we collaborate around the big data. The major challenge is, however, to stay granular with our data, even if it is a challenge by itself. Thank you.
Thank you so much, Stefan.
Yeah.
I'll invite you back on stage later on. I hope that you've started to think about questions you could pose to Stefan later on. I'd like to then invite our next speaker, Nilbert, who is the research director of the Danish Cancer Society Research Center and is also a professor at Lund University and University of Copenhagen. Take it away, Mef.
Thank you. So thank you very much for the invitation. Now to something completely different. I will talk about this region. I will talk about traveling from Denmark to Sweden and from Sweden to Denmark, and how we can perhaps do this a little better in the future when it comes to science, when it comes to clinical trials. I represent oncology. But this will be my only slide actually on oncology. In oncology, as you all know, I think the portfolio is strong in oncology. Precision medicine is the thing, genomic medicine not least. We have a strong pipeline, but we also need, I think, to really highlight the value of doing trials, of how we run trials in increasingly smaller and smaller subsets. It means we need to collaborate. We do see development of comprehensive cancer center infrastructures. That is happening in Denmark, perhaps even stronger in Sweden and Finland, also in Norway. These comprehensive cancer centers really focus on bringing basic science and treatment all the way through rehabilitation and palliative care together. We need to stand together between the research and the clinics. And this will also request and follow our inclusion into clinical trials, how we sort of manage and develop the systems to have more and more patients included. We are most of us below 10%. Patients in clinical trials, we know that the best centers in Europe can do it at between 30 and 40%. We also know that most strong clinical centers in Europe are struggling to get that high. This is not easy. Precision medicine obviously calls, as I said, for collaboration. I think that the Nordics can be an interesting market, but we need to realize that it's probably the Nordics rather than only Denmark or only Sweden. We need to include rapidly in clinical trials. We need to deliver what we promise. We have high quality. We have very skilled personnel. We have great data. We're not always quick. I think we can be quicker together. And we will see new endpoints. We will see not only one biomarker, but a biomarker panel, etc. It will be increasingly complex. So the best we can do to support both the developments in Denmark and in Sweden is to collaborate. We will also see patients requesting that. Patient involvement now is not just having someone at the end of the table, it is developing things together with the patient. So I will move on there, leaving the oncology area and the precision medicine to say that this is one of my sort of daily mornings. During the last year and going to work with this guy from India. He's a postdoc. Not many times I had to explain my exact relation with him and why we were traveling together in a car, etc. And that's only because he lived in Malmö, so it was convenient to go together. He was also quite afraid of those border controls. It is a bit difficult to keep this innovative mind up when you reach those barriers. But I included this picture to say that we have the Gränshinderrådet that are working with all these things. I think we should leave the barriers. I'm very tired of barriers. I think we need to look only and discuss possibilities. And we also need to think about which stories we tell. What is the image we provide? Is it that it's so difficult between Denmark and Sweden, we haven't integrated those tax rules and all of that? Or is it that this is a fantastic possibility? It's 20 minutes across the bridge and we are in another country. And we can really, with such a biotech cluster, such strong hospitals, strong universities, good science, we have all it takes. So I think that I included barriers to say, not one more evaluation on barriers. Let's move ahead together with the possibilities. And there is a number of possibilities. This is a picture from a recent report from Öresundsinstitutet. You should read it. It has so many good stories, and this is just one of the pictures of all of these people working together in everything from the bridge to science to clinical trials to you name it. There is so much good things ongoing. We should just also within our area make sure that we sort of hook on these developments. I think in this region that we have actually defined our strengths. In many clinical areas, especially perhaps the rare cancers, the rare diseases, we have strong Nordic relations. Childhood cancer, sarcomas, hematology, coagulatory diseases, there are many, many, many more. But maybe we've lost a little bit on that when it comes to the common diseases. But with precision medicine, common diseases will be increasingly rare. That is, the subset will be increasingly rare. So I think we need to re-establish those Nordic collaborations if we don't already have them. We have in this area definitely, as I said, high quality research and healthcare. We also have the processes to do really good clinical trials and to support innovation. We have great, actually, we have the biotech clusters, we have very good support in innovation, not least in the universities, also actually in the hospitals. We have fantastic clinical trial support, better than in many other countries. We will see more and more expert centers. We have defined expert centers, skill centers. We will see the comprehensive cancer centers growing up in my area. Then we have some points at the bottom. Now, I said we shouldn't talk about barriers, but I'm just going to say these are things that we, when we come into Europe or even to the US, say, we have great accessible health registers and biobanks, and we have efficient approval and implementation of new drugs. Well, I think compared to, for instance, the US, we are about to lose it. If we are not making our data and biobanks easily accessible, and if we are not more efficient in implementation, we will have a problem. Or maybe we will not have a problem, but we sort of missed the train, because the train has already left the station, at least in the US, when it comes to use of health data. To the right is just sort of when I think about the Öresund region and where are we strong, these are the sort of things that come to my mind. There are, of course, also others, but I think we have defined our strengths and we could profile those even more. After all of this, barriers and COVID, I think that we are at the time now, perhaps a little bit of a unique time where we do see a restart. We see a restart for the labor market. There is more awareness now that we need to collaborate. There is political collaboration, not least across the Öresund. We see increased traffic on the bridge and in the tunnel. We see a very strong and interesting infrastructure plans. And at the same time, we have all of these new research tools and possibilities. So I think this is the time to say that we learned something, we have new energy, but we also know that it's vulnerable. We don't take it for granted anymore that we can easily travel across. We appreciate it every day. So this is the time to sort of restart the collaborations and forget about the barriers. So this is my last slide. I sort of thought, what is it we really need? What is it we really need to change? I think that we could work with social sustainability. What makes it worth living and working and wanting to stay in this region? That should be very clear. We should have relationships. We should have support that support what we want to develop. which is healthy and livable communities, we should think about what are the needs for people who live in this region in 10 or 20 years, rather than focusing on those barriers, which we have hopefully now sort of left behind us. We should connect people. I think we need to start much earlier. We need to start in education. It needs to be easy to have trainees across the Öresund. It may exchanges from all different school levels into high schools and into university. Should be mandatory if you live in this region. It should be easy to exchange. It should be easy to take courses. I know that the faculties and the universities are working very intensely on this, but I also think that we need to work on it on all levels, because there is a strong willingness on the top. But unless you sort of get down to the lower parts where people are actually supposed to develop courses, etc., together, it's not going to happen. I think we need to tackle the challenges. One of them, obviously, running clinical trials, developing new drugs together, and realize that maybe it's okay, even if I'm Danish, to say that I give this trial to Sweden, I send 5 patients, so they get 10, instead we get 0 in Denmark, or the other way around, because that will in the end benefit us all. I think we should have more strategic decisions on which trials go here and which trials go there, and just show that we can more rapidly include into clinical trials. And then finally, what is the story? What is the story we tell? Is it that there are so many barriers in this region, it's hard to understand what they say on the other side, etc.? Or is it something else? I think that we all here need to think about what is the story I tell when I go to Stockholm or to Aarhus or to Oslo. This is a fantastic place to be. We have the position to lead. We can make the points where it really makes sense to travel across the bridge. We have the public support. Most people from Skåne love to come to Copenhagen, and they do that much more often than they actually go to Stockholm. We have a perspective. So the joint vision and making sure that the story we tell on this region and its possibilities for innovation is a positive one. Thanks.
Thank you, Mev. Appreciated your last slide, especially. I hope we get a chance to delve into it a little bit more. Our third speaker, before we go into the joint Q&A, is Marianne Pilgord. Marianne Pilgord is the CEO of Trial Nation, and you come from more than 20 years of experience with Novo Nordisk before joining Trial Nation. So welcome on stage, Marianne. I'll leave you to tell us more about Trial Nation and how it contributes to innovation in the region.
Thank you very much. And I feel so enthused and uplifted by the last speak, and I would like to continue on that, on possibilities. And I agree completely. I'm also one of the ones who go from one country to another, and let's celebrate that we are now past all these inspections. So, Trial Nation, we are a part of the clinical research ecosystem, and I will cover a bit about what we do, and some opportunities, perhaps some barriers for cross-border collaboration, and some ideas which I would like to bring to you, which we perhaps could work on together. So, for those of you who by any means do not know Denmark, we are quite a small country, 5.8 million people with outstanding clinical competencies. And just like many of the Nordic countries, we have extensive, saturated, longitudinal health data, and data in general, which has also been covered by other speakers earlier today. The Central Person Register, the personal identification number, has been in place since 1968, and we have electronic medical record systems, which are linked from cradle to grave. And we also have a national biobank with 22 million CPR-linked samples, and that's growing. And as you may know, cancer is one of the areas which is collected in this in the first round, together with 16 other areas. Now, Trial Nation. Trial Nation, we are a Danish publicly funded non-profit political initiative, and we pursue the benefits of clinical trials. And if you want to say in short, we relay stakeholder perspectives and we connect business with clinics. What we do is that we maintain and develop disease-specific networks who have excellent track records of industry collaboration. This we do in collaboration with all the 5 Danish regions. We address barriers to running clinical trials in Denmark, and we promote Danish clinical research capabilities, such as today, but also together with the Foreign Ministry towards different stakeholders across the globe. And we also inform about the synergies of clinical trials. And we really do believe that it's really in the benefit for all of us if we have the possibility of participating in clinical trials. We also have a vision, and our vision is that we pave the way to a future where patients enjoy stronger, sustainable health driven by fast access to cutting-edge medicine and medical technology, and where healthcare professionals save more lives and promote stronger health by always having state-of-the-art medical research and the latest discoveries right at their hand. And through clinical research, we create a better health and wealth in Denmark. And I think that the theme we have heard throughout this day, it resonates really well with what we are saying here, because we're all in this together, and together we can create a better whole. So, opportunities and barriers for cross-border collaboration. Currently, we relay feasibilities within all disease areas to all Danish clinical trial units. And in addition to that, we also support approximately 40 departments financially. That's actually regional money, which is redistributed through Trial Nation. And we also develop several tools to increase equal access to clinical trials. And again, we think it's a right for patients to know about the possibilities there are to participate in clinical trials. Currently, we are working on making a database available so that you as a patient can find, if I have this condition or if someone close to me has this condition, what are the treatment opportunities in a clinical trial for me, and where can I turn to in order to do that. And also for researchers to make more available what trials they are recruiting patients into. We really see an opportunity for attracting clinical trials to Denmark, but also to Sweden and other Nordic countries by the strength in numbers. Again, Denmark 5.8 million, Sweden approximately 10. All in the Nordic area, a small mega city. So we really do need the strength in numbers in precision medicine area. So what could we do then? Some ideas that I would like to bring to this. Well, we already have investigator networks. For instance, within Trial Nation, within the oncology area, we have the Nordic Early Cancer Trials, who already collaborate on a Nordic basis and are very willing on having that. And the physical distance in our particular region makes some of the barriers much, much smaller. We also have a pediatric network, which is organized both in Denmark, but also in each of the Nordic countries, and where we have very close collaboration across the Nordic area. So the clinical trials in rare diseases, and I think it has been said several times today, and Matthew mentioned it just in the last talk. Actually, most of the pharmaceutical inventions that we create today, they are actually in a rare disease, so to speak. And making sure that we have a larger population base, it actually makes it more attractive to run clinical trials in our area. So the cake becomes larger for us all. And it's also a very good way for us to leverage the data that we have. The fact that we actually have not only health data, but also supplementary data on all aspects of life which increasingly becomes more and more important in having access for the interventions that you design. And with that, I would like to close and say thank you.
Thank you, Marianne. Can you stay on stage?
Yes.
Because we're going to invite the other two speakers, Mev and Stefan, to join me. And I think, what we're going to talk about now really ties into some of the earlier discussions where there was a really big interest to see much more clinical trials, clinical research happening across the border and in one integrated region perspective. So how can we actually make this a reality? I mean, Mev, you were very passionate. We should be able to do clinical trials cross-border, offer one-stop shop. How can we make it happen? What does it take to see this come to reality? What do you think? Maybe start with you, Marianne.
Well, I think that using the already existing contacts, selecting some balloons, try balloons on collaboration, where the communication already is existing and where you have the relationships in place, to iron out the administrative barriers. From what we have seen, it's mainly administrative barriers, and that's much easier to resolve if you don't need to also establish a relationship. In areas where we know that there is a high throughput of trials, a high patient need, and we know that the patients really are willing to participate in trials across borders. We see that already today, but we see it more on an exception basis. Yeah.
Do you see that as a concrete way forward, Mev, or even Stefan for the...
Yeah, I mean, I would start by the end. I would say I agree. It's not the patients that is the problem. They would easily come across the region. So that's not it. But I think that maybe hospital leaderships, just as you say, could sort of say, let's look at 3 trials from Denmark to Sweden and 3 other trials from Sweden to Denmark. And let's try that, a pilot of 6, whatever, 8, 10 trials, and use that to sort of map the things that we need to just get rid of. They don't have to be very large trials because it's sort of conceptual, and we can use the areas of strength we have on both sides. Then I think it also comes down to relations. I mean, that's why I talked about education. You need to get to learn each other. Hematologists in Copenhagen should know the hematologists in Lund, Malmö Lund, and the other way around.
I think we should sell our product jointly as a trial area. And I think one major misunderstanding is that it's about providing the trial at the lowest price per patient, and that has created some despairs. Because the other nations could do that at lower cost, but I think the major driver is to have excellent data and to provide it with short notice. So to be very swift in the regulatory part and to produce the data, have enough patients to get the study to an approval state, because that's where the money really are. If you when you have a trial going on, you are on your patent already. So if you can provide the base for the regulatory approval quicker, it creates, I mean, it's billions of dollars for 1 year of delay, and that's our market share. That's where we can compete. That's where we should sell ourselves as providers, not having the lowest cost per doctor or nurse. That doesn't really make a difference in the end.
So ease of access, flexibility, good quality data. Speaking of data, we actually have a question here. Do any of you have suggestions for opening up the biobanks that we have in the region to companies and R&D programs in a better way than we have today? Because we often talk that we have great biobanks, great cohort studies, great patient registries. How can we use them even better?
Yeah. I want to, if I may, I want to ask people in this room, how many of you know about the initiative of one entry to all the registry data, which has been launched recently? Hand up. A few? much more could benefit from it. It doesn't say that you get free access to all the data, but at least you know that there is a one way into the system, because that's also one of the signals that we get, that it's very confusing, and with whom do I talk about getting access. And they've started the early first model, and it's operational.
Any reflections as well?
No, I mean, the trend is there. I mean, more and more journals do actually require you to give access to your data, and I think that comes for our big databases too, to make them accessible. And we are on our end at the Skåne University Hospitals working on data warehouse. I did that in US, anonymous data warehouses where you can actually get the data quickly without any extensive approval procedures, because they are anonymized. That's the way I go for the future, for AI, for any kind of trial you would like.
I fully agree. I mean, you need to anonymize it. Otherwise, we're going to be stuck in lengthy GDPR processes, which we already are. Then you can, of course, discuss how do you anonymize tissue. And I think we need to, talking about the biobanks, we need to sort of, I think data tissue can be anonymized, but that can be discussed, maybe if there are any lawyers here.
So the way we did it was by using honest brokers. So people outside the system can manage those data in a way where to the researcher it's anonymized.
But that's what we would call pseudo-anonymized, and that is not always, or that is not supported to be anonymized in our systems. But
I cannot talk about the regulatory issues here, but
you should be happy for that, actually, because that is a mess.
Yeah, but US, I mean, I spent so many hours with lawyers, and I'm fed up with lawyers in the US. But in this instance, there is a federal regulation that states that to be harmful for the patient, you have to be able to identify the patient. So by definition, if you anonymize data, and then of course there are some fields that are considered as identifiers. If you anonymize the data, it's considered non-human subject research, and then it doesn't work here. But I think we should work on getting it there.
And
I agree, but it's going to be very interesting to see how this plays out. But this is the way to go.
Yeah.
This is a hot topic, because I mean, you know the US environment, but GDPR is the big thing here. And actually, there's a question actually to you, Marianne, and maybe you can shed some light on this. Doesn't GDPR pose a challenge to the National Biobank with linkages between biosamples and CPR numbers? Does it or does it not?
Actually, I'm very happy that I'm usually not involved into that area. So I'll pass and send it over. Yes?
No, but I don't think it does. I mean, basically, we can share data within the EU. That is not so complicated. Only it takes a lot of time to make all these agreements. So if we could do it anonymized, but I don't see that as a problem, as long as you're sort of within the system.
Okay.
But this whole area, as you see, we need to move more rapidly.
Absolutely. And there's also another question. I mean, if we think about the European context, and I'm not sure if this considers, but one question was to ask, what can we learn from other innovative regions that span country borders? For example, the Basel area, and how? I mean, are they have the same constraints or not? Any inspiration that we can draw from there to here in Denmark and Sweden? Are you familiar? Any examples?
Yeah. I think that we have the very viable company structure in common, I think. And I haven't myself worked in the region, but being Swedish, actually from the Stockholm area and moving to this area, being half sort of Danish as well by marriage, and by also having very close ties to Denmark. I think that we have more things in common and more fruitful things, but I think that we focus on making it perfect. So I think that sort of lifting what the problems are rather than looking at the strengths.
Like you're saying, looking at possibilities more than the
It's also about being responsible for how we profile, what we tell about this area, this region.
Stefan, you were saying?
No, I just say that perfect shouldn't be our worst enemy. So, and of course, when we sell our region, we should sell the opportunities, not the difficulties.
And I actually have a follow-up question to that, Stefan. I mean, you're in a very interesting position in that you just came from the US environment with all these touch points, and now you're in Skåne. What has struck you the most, changing positions here? And what do you think we could learn from the US environment that we could apply in our context, knowing that it is really different when it comes to, spurring more clinical research collaborations or clinical trials in general?
So one thing, what I learned by doing it myself, to build the data in data warehouses, that was very fruitful in everyday clinical routine. The data warehouses were used with dashboards for surveillance of clinical pipelines. My own area, like intensive care, we had the ECMO service with 100 ECMOs a year. You could see on dashboard the leg complication. All that is only available if you have the big data in place. And so I think that should be one of our main focuses, to get our data in shape and place and synchronized so we can easily cross-reference the data across the borders. That maybe I'm biased. I'm talking about data all the time, but that should be one big facilitator for clinical research for sure.
Do you see that trend also happening in the Danish side, where more emphasis on such
Yeah,
structures?
I mean, I think we have quality registers. They are increasingly coming up as dashboards. So I think we're sort of on the way there. Probably not where the US is, because they were there 10 years ago. But we're moving there. But I would like to say that, I mean, look at what's going on in Germany and France and Spain when it comes to profiling strengths and centers and making them top European centers. If we want to do that, we need to collaborate more. And in my vision, hope, if the Öresund region could be one such top European region or sort of healthcare and research provider, that would be fantastic.
So what would be the top three points that you would raise in such storytelling exercise about the region that you think would really capture attention?
Probably be our key areas we are strong at, this and that. We can deliver on clinical trials and research, which we actually only can, but we need to show it. And maybe that, this is the socially sustainable region. This is where you want to work and live and have your kids and see them grow up to become scientists or doctors or nurses.
I'm a cross-border commuter myself, so I identify with all that we're talking about here. I wanted to talk a little bit about the value of clinical research. We're talking about this innovation in clinical research, in improving patient outcomes and so on. But we know that it is really difficult to get clinicians to prioritize it day to day. I'm not sure if this is unique to the region. I think it's everywhere. How do you think we could help clinicians better put clinical research on the forefront and really take advantage of all the ideas and innovation that they have?
So being a clinician, I would say that every clinician would love to do that. This is what you should do when you work in a major hospital. So there is nothing, the barrier is not in the clinicians, the barrier is in the leadership. The leadership needs to prioritize this and then realize that it takes some more time. You need to have time set apart. So this is a leadership issue in my perspective.
But also resources. So for instance, I learned from US that you don't need study nurses to that extent that we use here. So we had study coordinators. They cannot deliver drugs. They cannot take any intravenous or intra-arterial samples. But all the other regulatory issue consenting some can done. And this was kind of a lucky shot I did. So when COVID came, we could just continue. But here in Sweden, everything was stopped because all the nurses were pulled into the COVID. And this will not be the last pandemics we see. So I think we need to safeguard that research can go on. That's one thing, and I would prioritize. And then I would also say that it's not on the doctors. They, in many times, they want to do it. I would say, if you ask me, and I put your question out on the floor here for you, what would be, you have one wish, Stefan, and I would to streamline the regulatory issues. So that I think kills most of it. That regulatory issues are unnecessary, lengthy.
Okay. But would you say that indicates as to the rigor and it adds quality to our clinical trials to have that regulatory perspective in place?
Yeah, I think it could be made more efficient by have predefined paths for similar trials. And then it doesn't need to be reinvent the wheel every time you need to get it approved.
We also have a question that is kind of a bigger picture perspective. I mean, we are in 2021. We know we've had quite a crazy past 2 years. We know that COVID has really impacted clinical trials in many ways. This question is more towards, do you think the pandemic and vaccine development will impact the interest of patients to participate in trials going forward? It's quite a, what are your reflections there?
I mean, I would say, in general, this will sort of support science broadly, demonstrate how important science is, and also showing how rapidly we can take things from discovery to implementation. That, I think, is the most inspiring and energetic question, which means that if you discover a molecule, you can actually take it maybe to a drug with mRNA principles, etc., in much shorter time than we did. We've already seen this happening, so you need to, but that also means that you need to sort of be ready for the future. You can't just sit there and say, oh, there was a drug coming out now. How am I going to implement that? So it also sort of pushes some needs on us on the planning and horizon scanning.
Yeah, and I agree. I think that the past year and a half or 2 years has really shown what a vulnerable society we are if we don't have the medical innovations. And just put ourselves 2-3 generations back. I mean, that was really the case back then. Very limited treatment interventions, and this has really shown what can be done when we collaborate together.
But also, the population in Scandinavia is very put a lot of trust into science. I must say it's that has taken a turn in the later part of the pandemic. Being a dual citizen, I love United States and so on, but it's been a question around this. The skepticism in US with the most horrible ideas, or even being delusional ideas among 20% of the population. So, it actually impacts the power of science because you cannot get it out to the population. So, we have great opportunities, even with regard to the trust that people put into science here.
And perhaps that's one of the things that we can sell about our region, in that the Nordics generally have a very strong appetite to engage in trials compared to, say, the US and elsewhere.
I'm just not sure we should take it for granted. Other things from the US have been imported to us.
Yeah.
So.
No, I mean that any kind of Macaroni scandal will actually take a toll on the trust of science. Sorry for bringing it up, Stefan.
We also know that COVID has, transformed clinical trials, previously very, location dependent. Now it's moving more to a discussion on virtual clinical trials or remote clinical trials. Do we see that, as this is a question from the audience, do we see that that's a mean for collaboration between Denmark and Sweden? A way that we can join forces?
I would definitely say yes. I would also say that this is a wake-up call for the big hospitals, that they need to think about the regions. They have responsibility for a network, and this network can easily extend across the border as well.
I also think that the regions in US, people are reflecting on that within 10 years, 150 institutions will cease to be because of the competition. We need to collaborate, but we need also to focus on the power centra we have. In Sweden, there has been a diversity to spread out universities in every little small city and resources to every little small city. We need to see, so for instance in Region Skåne, we need to see that we have a Skåne University Hospital. We need to focus to get Skåne University Hospital to be able to do this international collaboration. So to actually a power focus also in Denmark on the big academic centers.
I think also that the patient perspective is very important here, and the treatment burden on a clinical trial. The more you can decentralize, the easier you make it, both for patients who live far away, but also staying in the trial and doing all the procedures which are needed for an extended period of time.
I think the drugs are increasingly also on per oral drugs, low side effects, many of them, and they can or should move out to home-based trials, etc. So that's why you need to think about the region.
Yeah. I have one last question from the audience that I think could be a very interesting way to wrap up. So this person asks all the speakers, if you had unlimited resources and money, what would be the first thing you would do in this region, I guess, to foster more cross-border collaboration?
Unlimited resources. Does that also mean that people will automatically say, want to do everything that you suggest?
It depends.
I think that having the possibilities of having research as seen as an integrated, necessary part of your clinical practice, I think that would be a baseline. And then I would actually have a task force to work on smoothing the barriers, the generic barriers, which we presume will be in all the cases. I think that what MEF presented in making sure that you have the ties, the connections between the clinical environments to provide an arena for that as well, to foster the collaboration, I think that would be the sort of way to go.
So partly, I don't have all the money in the world, but we are on the Swedish side in the restructurization, in the sense that we are building all the new hospitals in the major cities, and so Lund gets its news, Malmö gets its news, Helsingborg. And what I think is take that opportunity to embed research within the hospitals, to make the distances short, make people aware of each other's and joint databases. That would be what I would focus on.
So, I mean, I'm sort of in the same ballpark. I think that clinical collaborations, clinical trials, or research takes people. It's about people who want to collaborate. It's not something you can sort of structure, you can build. You need to make people meet. So I'm sort of back to the make sure that people exchange, maybe take away the toll fee for the bridge or something if you have a lot of money. Make sure people can just...
You have a lot of money.
So I take away the toll fee.
I will support that.
But not because that's the barrier. But I think that people need to meet, and you need to start early in school, through the educational system, and then research. Maybe require that for grants.
Yeah,
from
really
this
good.
region.
Excellent. Very good ideas to bring away with. So thank you so much for your time,
Thanks.
Marianne, Stefan, and MEF. All right, and now for the presentation I think a lot of us have been really looking forward to. It is really my great pleasure to invite Dr. Robert Langer, who will anchor the rest of the day for us. He's joining us live from Boston. And while preparing to introduce Dr. Langer, it was a bit intimidating, I must say. You have a really strong list of credentials, but just some numbers here. Dr. Langer is one of the 12 institute professors at MIT, an institute professor being the highest honor that can be awarded to a faculty member in MIT. Dr. Langer has written more than 1,500 articles. He has over 1,400 issued and pending patents worldwide. He is also the most cited engineer in history, with over 355,000 citations, according to Google Scholar. So, Dr. Langer, fantastic to have you with us, and I'd like to encourage you here. Make sure you send in your questions on our Q&A platform. Dr. Langer, we have about 300 people here in the room. Looking forward to hear you share with us on your thoughts on creating and implementing breakthrough technology. So take it away, Dr. Langer.
Well, thank you so much. Can people hear me okay? I'll assume you can then. So what I was asked to talk about was innovation. So I actually made a slide, and then I'll try to back it up. If we could go to the slides, please. If we could, yes. So I sort of put the 6 things down, that I thought were important, at least to me, in innovation. One is, and this is what I also tell my students, when they go to companies or academia, first do things you hope will have a very big impact, as opposed to just what I'll call incremental research. Secondly, conventional wisdom, which is what you read in books and everything else and what everyone tells you, is not always correct. Third is really multidisciplinarity, exposure to very different disciplines. Fourth, take risks. Failure's okay. And five, if your idea is important, you should probably expect lots of criticism. And finally, even when you get all that criticism, don't give up. I mean, at least not for a long time. At least this is what, and I'll try to back that up in terms of my own experiences. So when I got done with my doctorate, it's kind of a long story, but I ended up, even though I'm a chemical engineer and I got 20 job offers from oil companies, that's what chemical engineers did in 1974 when I finished. I got actually 4 from Exxon alone, but I really wasn't excited about those jobs. I remember going to these interviews and, one of the people at Exxon said, gee, if you could increase the yield of this one petrochemical by 0.1%, he said, that would be wonderful. It'd be worth billions of dollars. And I remember flying back to Boston that night thinking I didn't want to do that. So at any rate, I kept looking for ways to use my chemical engineering education to help people. And one day, I remember walking into the lab and one of the older scientists said to me, he said, Bob, you should write a surgeon named Judah Folkman at Harvard Medical School and Children's Hospital. He said, sometimes he hires unusual people. He thought very highly of Dr. Folkman. I won't say what he thought about me. But anyhow, I wrote to Dr. Folkman. Dr. Folkman had this idea shown on the next slide. And without going into a lot of detail, what this next slide shows you, if we could put it on, please, is that if you start on the left, you have a tumor cell, and it behaves abnormally. It grows as a 3-dimensional mass. And when it gets, if you look to the middle bottom, it maybe is 1 million cells, but then it runs into a nutrition problem. It can't get nutrients in the center or get rid of waste. But the way the tumor solves that problem, he said, is it secretes a signal, which he called tumor angiogenesis factor. And that solved the nutrition problem for the tumor, and it started growing and growing. And if you go to the upper part, it's very vascularized, and it spreads through those blood vessels. And when it metastasizes and does that, that's often what kills people. He had an idea, and it turned out to be widely criticized. I didn't know that because I didn't know anything about biology. But anyhow, I thought it was a great idea. His idea was that if you could stop blood vessels, achieve anti-angiogenesis, maybe that would be a whole new way of treating cancer. So one of the biggest problems is we didn't have any way to study blood vessels. And that's often the case when people come up with new factors. So we had to invent one. And so the next slide shows what we tried to do, that we could put certain tumors in the eye, but then what we needed was a delivery system that could deliver any of the molecules we were isolating. And they're all very large molecules. So the delivery system had to be safe, and it had to be able to deliver these molecules. But Dr. Folkman was on the board of the one company in the world at the time that was working on this, a company called Alza. And he went there and he talked to a number of people, including Nobel Prize winners, and they all told him this was impossible, that you couldn't get large molecules to be delivered through solid materials. They were just too big. It's like walking through a brick wall. In fact, the next slide just shows what some people wrote even, that large molecules can't slowly diffuse through solid. I'm sorry, if we could go to the next slide. Yeah, that the use of polymer matrices for slow release systems has been virtually restricted to small molecules. But if we were going to solve the angiogenesis problem, we had to do this. So I actually spent over 2 years in the laboratory experimenting with different techniques. I found over 200 different ways to get this to not work. But eventually I was able to make little micro or nanoparticles. Next slide. You see one on the left and cut in half on the right. And we published in the British journal Nature, next slide, in 1976, that you could use this approach to release molecules of any size. In fact, this was, to my knowledge, the first time anyone had ever also delivered nucleic acids from tiny particles like nucleic acids or DNA or RNA. Nonetheless, when I first presented this work, and I was 27 years old, I presented it at a scientific meeting. I thought I was, I practiced this for a long time, and I thought when I was done with the talk, I didn't forget too much what I was going to say. So I thought all these older scientists, being nice people, would want to encourage me, this young guy. But when I got done and stepped off the podium, a whole bunch of them came up to me and they said, "We don't believe anything you just said." They said, next slide, that that just what the ALSA people said, that the molecules were too big to get through solid materials. And we also were using organic solvents, and they said that would destroy whatever we put in, peptides, proteins, or nucleic acids. And actually, unfortunately for me, things went downhill from there. I tried to get research grants. My first 9 grants were rejected very badly. I remember the reviewers at the National Institutes of Health writing back on a cancer grant I wrote that Dr. Langer should not get this. He knows nothing about biology and even less about oncology. But I thought I knew enough about math to know that wasn't possible, but I wasn't, calling the shots. So that wasn't very good. Then, I really liked being a postdoc, but a lot of my friends said, well, it's probably not good to be a postdoc forever. So I started applying to faculty positions in chemical engineering, which is my discipline. But no chemical engineering department in the world would hire me. I like to think the reason was they thought that the biology stuff I was proposing at that time, didn't fit. I mean, maybe there were other reasons, like I didn't interview well. But anyhow, nonetheless, I didn't get any jobs. And so finally, Dr. Folkman knew a man named Nevin Scrimshaw, also from that ALSA board. Dr. Scrimshaw was a very famous nutritionist in charge of the nutrition and food science department. And he was also what I'll call a benevolent dictator kind of department head. And he offered me a job. But the thing is, he didn't bother to ask anybody else in the department what they thought, which might have been okay, except for one thing, which was that the year after I joined the department, he left. So a lot of the senior faculty decided to give me advice, and their advice is I should leave too. Well, anyhow, I'm still at MIT, but, it was shaky for a while. At any rate, as I mentioned to you, one of our goals was to see if we could use this to isolate and find the first angiogenesis inhibitor. And we did that. I'm going to show you a bloody slide. There's no other way to show this. From a paper Dr. Folkman and I wrote in Science in 1976. And we tested literally 100 fractions, 99 of which did not work, but one did. And let me go to that next slide, which is bloody. Oh, I'm sorry. Yeah, this was what I was going to say, maybe just to show the last slide for a second, this was actually one of my friends, Mike Marletta, saying that, talking about that people wanted to get rid of me. And if you read this, he said, when the older scientists heard my concepts for drug delivery, he blew a cloud of smoke in my face and said, you better start looking for another job. But at any rate, let me go to the next slide. This was the slide that I wanted to talk about. And what you see on the left are all these blood vessels growing to the tumor. It's about 8 weeks after the start of the experiment. We did this probably about 2,000 eyes, 100 fractions. On the right, though, what was striking is we show that with the inhibitor, the one thing we found that you see blood vessels sparser, lower, and they actually don't grow into the tumor. And shockingly, 100% of the time, the tumors on the left grew. We sacrificed the animals generally before that. But on the right, 60% of the time, they never did. And so this proved for the first time there were blood vessel inhibitors, and it would also establish techniques by which people could isolate all future inhibitors. Now, it took 28 years from this Science paper before the first angiogenesis inhibitor was approved by FDA. But let me show you what happened starting 28 years later. Next slide. What you see is one after the next angiogenesis inhibitor approved by FDA. Avastin is the number 1 or 2 best-selling biotech drug in history. And not only have they been approved for all kinds of cancers affecting tens of millions of patients every year, but also these are the only pharmacologic treatments for treating blindness in the back of the eye, diseases like macular degeneration or diabetic retinopathy. These are drugs like Eylea or Lucentis. These are also blockbuster drugs. Now, Dr. Folkman told me one day, we were talking, by the way, I was the only engineer in the entire hospital when I worked there. But Dr. Folkman and I talked one day, he said, well, Bob, we should file for a patent. Now, when you hear that I have like 1,400 patents, maybe you think that's normal. I had none at that time. And Children's Hospital, a very good hospital, they didn't have any either. So for the first time, we filed for a patent, and 5 years in a row, it was rejected. So the head of the tech transfer office at the hospital came to see me one day, and he said, Bob, stop. He said, you are wasting so much money that patent will never get allowed because of the examiner. He doesn't understand science. He said, just quit. But you can probably tell by now I don't like to quit. So I started to think, how could we convince the examiner to allow this patent, legally, of course? And science wasn't working. So I wondered, maybe somebody wrote down that something like this. So in 1982, 6 years after we published our Nature paper, I looked back to see if anybody wrote about this. And on the next slide, I actually, hundreds of people have actually, or thousands, have written about it, but I'll just focus on this one. And this was a paper from 5 of the top material scientists in the world, and they're saying generally the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules could be released by such a technique because of their extremely small permeation rates. However, Folkman and myself have reported some surprising, so surprising is a really good word for a patent examiner, results that clearly demonstrate the opposite. So I showed that to the head of the tech transfer office. He flew down to Washington to show it to the examiner. And the examiner said, well, gee, I had no idea. He said, I tell you what, I will allow this patent if Dr. Langer can get written affidavits from these 5 people that they really wrote that. So I wrote them, and they were all nice enough to write me back they really wrote it. And we got this very broad patent allowed. Next slide. And with that patent, we actually licensed it to 2 very large companies, which gave me grants and consulting fees. But unfortunately, they were both multi-billion-dollar companies, one in animal health, one in human health. But unfortunately, they didn't work very hard on it. When the first experiments didn't work, they gave up. So a couple of years later, I got the patent back, and we started this little company, Enzatech, next slide, with 4 of my students. And today, that company is worth about $5 billion, has 25 products approved or in clinical trials, new treatments for many diseases like schizophrenia, opioid addiction, type 2 diabetes. And we also taught other companies how to do this and did other licenses. And the next slide just shows a bunch of pictures that are of all these products that are now FDA approved. They range from little microspheres that you can inject underneath the skin to treat cancer. All drug-eluting stents are based on this, and so forth. So, but we continue to make discoveries in our lab, and again, we would patent them. And again, I found if I'm not my own champion, nobody else will be. So let me give you 2 more examples. The next one, if we could go to the next slide, are biomolecules. You know, the people have come up with proteomic companies, which their proteins are all monodispersed. Nucleic acids, those are also monodispersed. But polysaccharides, they're much more complex. They're polydispersed, right? Heparins are polysaccharides, and they are really a mixture. So what we did, if you have one of these mixtures, next slide, is that you could fractionate the mixture and find what we call glycoforms. There are probably thousands in there. But what we did, which nobody had ever done before, is sequence polysaccharides. We did this based on 2 papers in Science that my postdocs, Ram and Ganesh, wrote, and myself. Basically, for the scientists, it's a combination of molecular scissors like heparinases, cutting these at different places, then using mass spec, and then developing algorithms that could put the cuts back together. And so what we did is we thought you could make different kinds of products like are shown on the next slide. Heparins, other complex polymers, or new glycoproteins. So in 2001, next slide, we started another company called Momenta with my 2 students. We got venture financing. We went public in 2004. And then what we did is we'd give you have to finance the company, so we'd give little pieces away. This is like a platform technology, meaning you can apply it to different things. So we did deals giving little pieces away to Novartis, Baxter, Mylan, and CSL. And that's a win-win for the big companies, which would get rights to the products, but also for us, because we could do more research. And what happened is, even though there were many larger companies working in these areas, trying to, say, sequence or come up with different molecules, Lovenox, which is a multi-billion-dollar product, that actually was worked on by Teva and others. And all the stock analysts always said they would beat us by a long shot. By the way, they still haven't gotten FDA approved in 2021. But in 2010, we did. It was the first Lovenox, the first complex drug approved based on analytic data. It's the largest syringe launch in history. Then Copaxone was approved in 2015, and Johnson & Johnson acquired our company in 2020 for $6.5 billion. Just to show you again some pictures, next slide. This is enoxaparin, that's low molecular weight heparin. Of course, the company did well when this got approved. Next slide. But I wanted to tell you one last story about the good these companies can do. In 2008, next slide, you may remember that there was contaminated heparin all over the world. And I, at one point, was also chair of the FDA Science Board, so they contacted me and said, well, could we help figure out where the contamination was coming from? So we at MIT, myself and Ram Sasakaran, our scientists at Momenta, along with the FDA and the CDC, Center for Disease Control, published three papers. Next slide. And these, on the bottom, we identified the contaminant, which was oversulfated chondroitin sulfate. And then we published two other papers in the New England Journal of Medicine going over what the problems were. And after this, there were no more deaths. Now, another basic discovery we made, and again, when we make a lot of these discoveries, it's not necessarily that we know where they're going to lead to. But I also had this idea of, as I mentioned when we did that Nature paper, we could make nanoparticles, but as well as microparticles, but those nanoparticles would sometimes aggregate, and also there were cells called macrophages which might eat them, even though the kinetics were okay. So we came up with a way to make nanoparticles, and I'll just show you a video of that, where we put a molecule called polyethylene glycol on them. This is a video from the TV show Nova. Next slide.
He starts with a nanoparticle of anti-cancer drug. That gets encased in a plastic that releases the drug over time. That in turn gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
So it doesn't really blow the cell up quite like that, but you get the idea. And we began, and others too, using these approaches to deliver small molecules. But what's really exciting to me is that you could deliver large molecules like siRNA and messenger RNA. And that brings me to the next point. If we could go to the next slide. In 2010, myself, Derek Rossi, Ken Chen, and Nubar Fayan had this idea that we could create a company to deliver messenger RNA. And you'd want to put the messenger RNA in nanoparticles, otherwise it would get destroyed. But let me motivate this for you. So protein therapeutics have become a huge industry by then. In fact, I think now something like 70% of the top best-selling drugs are proteins. But it takes a long, long time to make them, to purify them. And there's a lot of diseases you can't even treat with them, like intracellular diseases and membrane-bound diseases. If you could deliver mRNA, messenger RNA, the body would actually do that for you, right? Because DNA makes RNA, makes protein. Also, messenger RNA would be an easier delivery problem because all you have to do is get it into the cell. DNA, you'd have to get into the nucleus. So that was our idea. So we started building Moderna in 2010. And by early 2020, we had 13 products in human clinical trials. And then COVID hit. So then we started to work on that as well. But again, just like I was criticized early in my career, people may not remember this now that it's all approved, but when we started doing this, so many scientists and clinicians all over the world criticized us and me and said it would never work. In fact, let me just back that up with a headline from our local newspaper, the Boston Globe. This is the main newspaper in Boston. And this is actually, if you look at the date, May 19th, 2020, after we'd even started clinical trials, and all these people told the Globe, this is not how you do science. So that was the headline with my picture on the front page. So that was not too pleasant. But let me just show you what Moderna was able to do and the power of this technology. Next slide. So you may know that the Chinese scientists published the virus genetic sequence on January 11th. 2 days later, we finalized the design. pretty much just a little over a month later, we shipped the first batches. We made it and got the right nanoparticles, shipped it to NIH for testing. And we did the first dose in patients literally about 2 months after this all started, which was historic. And then by the end of the year, as I'm sure you all know, it would get emergency use approval by the US FDA and many European authorities. And it's now, next slide, used all over the world. And by the way, the BioNTech Pfizer one also uses nanoparticles to deliver it. So you know what, I thought I would try to spend the last 5 minutes or so going over what Jeff was one of my postdocs for many years, and now he is a full professor at Harvard. But he actually went through and analyzed what we did at MIT. And I think that in terms of these companies, so by the way, did Harvard Business Review, but I thought I would just take, he let me his talk about what we did, what I did, and he of course lived it. So I thought I would go over some of that, which I think will back up some of these points. So a lot of what we do has materials in it, but the materials can be used for different things. And then he took different interviews that I gave, and I thought I would go through some of those. Next slide. So this is me talking about working in Children's Hospital back in the 70s. I said being an engineer working in Children's Hospital was a great opportunity for me. I would see many medical problems, and I started to have a lot of ideas about how to approach them. It was almost like being in a candy shop. I had very different perspective being an engineer. Remember, I was the only engineer in the whole hospital. I could see how using chemistry could address this problem, or engineering could address that problem. I'd be riding up the elevator with clinicians, and they would say, well, we have trouble with this. And I would say, well, maybe we can solve it this way. And then as Jeff also pointed out, I had a hard time getting grants because my ideas didn't meet with conventional wisdom. The next slide just shows, points that out. And on the next slide, these were reviews that I got on this idea I had of a new material. And 7 times in a row, these grants got rejected by different reasons, because people thinking we couldn't synthesize the materials, or they'd react, or they'd be fragile, or they'd be toxic, and so forth, until actually finally the FDA approved it in spite of all of the reviewers' objections. Another thing that I said apparently in one interview, next slide, is I said so many times when you try to do something in science, when you try to invent something, people tell you it's impossible, that it will never work. But I think that's very rarely true. I think if you really believe in yourself, if you really stick to things, there's very little that's really impossible. Jeff also analyzed the types of people in our lab. Next slide. And again, the philosophy goes back to what I said in the beginning, to try to make a difference in the world by working on significant problems. But first of all, we are well funded. Today, we certainly were not in the beginning. We have gotten lots of patents. I think those patents have been absolutely critical. A lot of people criticize patents, but my feeling is, 99% of the discoveries people make, if you're not your own champion, it won't go anywhere. Every so often, somebody makes a discovery that may be so fundamental that people will just accept it. But most of the time, in my experience, people don't accept it. And investors might put money into it, but they won't if you don't have a patent in medicine. So we've done that repeatedly. Next slide. The lab is very interdisciplinary, over 100 people. And like I say, all kinds of different backgrounds, from medical doctors to chemists to veterinarians. Next slide. And again, we try to innovate, pick high-risk research problems. The people have passion, intellect, they like to collaborate, there's trust, respect. And like I say, we have fairly good funding. Next slide. So Jeff says my model of an ideal research product is a big idea. And then, as you might have known from what I talked about, and maybe this is me being an academic snob, but we've always published everything we do. And we try to publish it in really top journals like Science or Nature or New England Journal of Medicine. And then we patent it too. We do both. Also in medicine, things take a long time and investors get tired. So we want to have very effective in vivo studies, usually in animals. Next slide. And so another interview, I apparently said in my own laboratory, I let people see examples of what I do. I have people running around in here all the time about, can you patent this? Can you patent that? And I think role models end up being important. When people see other people do it, it increases their confidence and awareness of the way they think. And I like to think they're more likely to succeed. Sometimes people ask me, well, what does MIT do to help? Next slide. Well, one is I think MIT has lots of good role models. And that's one of the things we have an undergraduate research program that provides. It's a terrific way for undergraduates to learn research. And I think that helps getting the students in at a young age, learn research, and part of what makes MIT unique. But then I think MIT goes a couple steps beyond just education. MIT, even though I like to think we do very good research, we've also had very strong ties to industry. Like MIT has like quite a number of people in a technology transfer program, and they also have a big what's called industrial liaison program. And so those have outreach. And I think since we have all those things, they expose students and professors to a broad spectrum of things, and I think encourages innovation. And on the final slide, next slide, I think it's important to encourage people to reach for the ideas, to think big, to give positive reinforcement about discovery and invention, and to create an environment where people can do that. So I'm going to stop here, but really it's been an honor to present to you. I've always had such enormous respect for the research and companies in your region. We have actually had great collaborations with Novo Nordisk and still do. And I've had certainly lots of interaction with people in Sweden. And, it's, like I say, it's an honor for me to present to you. Thank you so much.
Thank you so much, Dr. Langer. You probably didn't catch it, but we in the audience really, really enjoyed your presentation. It was so inspiring to hear your journey of grit and perseverance. And we just wanted to perhaps start with just hearing your thoughts from where you stand in the US. How do you see the Nordics? Do we have the conditions that it takes to create the breakthrough technologies that you are describing?
I absolutely, I've lectured at many of the, I actually have 2 honorary doctorates at Sweden. I've lectured at the Karolinska and Uppsala and Lund and Gothenburg. So and same thing in Copenhagen. I've lectured there many times. And as I mentioned, we have had a great relationship with Novo Nordisk, where we're, we're co-developing actually a very interesting oral delivery pill for insulin and other molecules. So I think the research at the universities is terrific. And I think some of the companies have been terrific. So I certainly feel you have what I'll call the raw materials to do it. And obviously Novo Nordisk and Copenhagen, I mean, that is one of the world's great pharmaceutical companies. And I'd say that the Karolinska is one of the great medical centers in the world.
And I mean, in your journey, you really pointed to, really the perseverance that is needed to bring an idea to reality, especially when you're talking about transformational ideas that break conventional wisdom. Do you see that there are certain elements to creating or developing people that think in that way? Is that specific to the US, or is that something that Nordics can pick up? Like, what advice would you give to us given the culture that we have here?
Well, I'm hardly an expert at the culture, but by the way, I personally think that you all have done great. But what I think about is it really goes to some of the things I said on the very first slide. I mean, and by the way, and part of it's feeling that failure is okay. Taking risks. If you take a risk, you could easily fail. And some cultures are really failure averse. But I think the one thing that has been good in the United States is failure is very often accepted. I failed tons of times. Bill Gates has failed. Steve Jobs has failed. I think Elon Musk maybe even failed. I don't know, but but failure is an okay thing. And I think that some cultures there's almost a shame to failure. And I think the way to think about it is it's not about failure. And by the way, even some review panels, that I've looked at for grants, they say, well, this won't work because of this thing or that thing. But I think what's really key is to look at what the possible benefits are if you do succeed, rather than what'll happen, the likelihood that somebody will fail. So I think that's a part of it, anywhere. But my personal feeling is that people in the Nordic countries have done great research. Part of the issue is also size. I think in the United States, we have many more people. So, when you look at things, it's sort of easier. But I also think the other thing that's really important is to support, almost all the time that has led to these companies has been supporting basic research in either science or engineering at universities. I'm obviously I'm very prejudiced about that being at a university, but I really feel that the more funding that goes to basic research is critical, because you don't know when you do the research where it's going to lead. I mean, when we develop these things in in Nature papers and everything, we didn't know that, like I say, it was based on trying to help basic cancer research, but it would turn out to be very important for, COVID vaccines and other things we never could have dreamed of when we started that work. So I think, and then the next kinds of things I think are, and I'm not an expert on this, but there may be laws, like how do you, incent people to invest? I mean, that I think, because I know there are differences in, from when I've been over to Denmark and Sweden, that there's both differences to do philanthropy. There are better laws in the United States to encourage philanthropy. And then also, there's capital gains kinds of taxes for long-term capital gains kind of taxes for investments. So those kinds of things I think would also be probably very helpful.
But I think you're touching on a really important point. It's to normalize failure in some way, to talk about failure as part of the journey to success a lot more. And exactly what you have done. So that's really inspiring and something to take away. I actually have a question from the audience, and just some reflections from you, Dr. Langer. From your perspective, what are likely the next breakthroughs in the medical world, given the direction of latest innovation, success, and research? Where do you think the next big thing is happening in life science from where you stand?
Well, I've been asked that question before, and I can tell you areas that I think are important, which are cell therapies and regenerative medicine and obviously genetic therapies, like I've mentioned. But really, my answer goes back almost to my last question, but in a somewhat different way. I always say when I get asked that, it's something that doesn't have a name because it hasn't been discovered yet. And I really believe that. What's going to happen is people will make discoveries, maybe already have made them, but they don't have a name. And to back that up, if I asked anybody in the audience 10 years ago what CRISPR was, they would not know. I mean, and so I really think that people need to think about basic research that can lead to discoveries, and those kinds of discoveries won't have a name right now. But if those discoveries keep happening and inventions keep happening, it will make a difference. I'm just not smart enough to know what they are, because I don't think they've happened yet.
And, Dr. Langer, you were describing your team, and I think what really stood out to me, at least, is the multiple disciplinaries that are in your team that really facilitates this innovation that you're talking about. When we talk about the future, what kind of competencies do you think are going to be key for us to find these breakthrough ideas that we don't know yet about?
Yeah, well, I think that they'll come from really every area of science and engineering. I do feel like biology, chemistry, physics, math, I think artificial intelligence and things like that. But then I also think all the engineering disciplines, chemical, I'm prejudiced, and biological, but people are doing interesting things in mechanical engineering with computer science. So I think they'll come all over. And one of the things that Phil Sharp and I wrote in Science 10 years ago also, which relates to one of the other points I made on that first slide, is this idea of convergence, that you really want to bring disciplines together, unusual disciplines that may not necessarily fit. I mean, I could envision people in computation talking with meteorologists and coming up with new ideas, about weather and all kinds of things. But my point is that putting people together, and that certainly really made a difference in my career, being the only engineer in a hospital. I think that putting people together with very different backgrounds, it stretches you, it makes you grow, and it makes you think about things you might not have thought about before. And I think it also helps others. in the lab.
And I think that's a really good perspective, especially for life science. I think sometimes we're quite challenged to move beyond our traditional pillars of life science. So I think that's a really good word for us to take away with. So Dr. Lang, I just want to thank you once again for your time with us. And we look forward to perhaps welcoming you physically to the region sometime in the near future.
Well, I love your region. You probably have some of the best food in the world, in addition to everything else.
Exactly.
Thank
you, Dr.
Thanks
Great,
so
thank
much.
you. Thank you, Dr. Lang.
Okay, thank you.
What an inspiring way to wrap up such a rich day of presentations. And I'm not going to take too much more of your time. I would like to hand over the final word to Søren, our chairman at MVA, to wrap up the day and summarize and conclude for us. So over to you, Søren.
That's a tough one to follow, huh? So those of us who have had the honor and pleasure to actually meet and discuss with Dr. Langer in real life know what an energetic and inspirational force he is. It's hard to stay sort of in your body when you're with him. You want to do more with your life. It's just fantastic. And what a fantastic day we've had here today. And I'll not spend so much of your time because there's red wine, white wine, and tapas out there. First of all, we started out welcoming Annette as the new CEO of Medicon Valley Alliance. And Annette, if this meeting is sort of a testimony to how the next many years under your leadership will be, then it's going to be fantastic. So thank you to you, and not the least, thank you to the entire MVA team for putting this together. And I have Bob Langer down here with a very stern look up at me, so now I'm back there. So thank you for that. We started out getting reminded how important life science is for the region in terms of growth, wealth, and not the least health. Annette took us through the state of the Medicon Valley Alliance, and later the chairman of the Danish commerce organization, Brian Mikkelsen, reinforced not only the need for life science to keep prosperous societies, but also talking about the importance of collaboration in the region. And I think all of us that are here today can, of course, agree to that point. Then we had a little bit of a discussion about the difficulties or we touched on the point of the difficulties in translating all the good science that we have in the region, and we can agree we have that. But we have problems, difficulties, challenges translating that into innovation. And we had two talks, both from Matthias Olén and latest here from Rob, that must give us hope that it can actually be done. I mean, great scientists that have been able to take their innovation or their basic science, applied science, all the way to innovation and company creation. But we are not all like Bob. We are not all like Matthias. 26 companies, I think Bob have more than 50. That's probably not within reach for all of us. So what do the rest of us do? How do we get from being scientists in the lab doing what we like to become maybe single entrepreneurs or multiple entrepreneurs? And we had a very, very inspiring session with Jens and with Peter and Jonathan from the US, really talking about how do you set up your company, how do you make a business plan, how do you make it sort of milestone dependent, and how do you set up your dialogue with investors and eventually business development partners to prosper in this world. That was really, really important, and I think we will come back to that theme at MVA in the years to come. And the final track we had, and that ended up with, I guess, the most inspired and energetic discussion I've heard about the data, GDPR, and clinical trials for many, many years. That was really, really great to follow by Marianne, Jonas, and Mev. Great perspective. It was started by Lynn, who gave us perspectives on AbbVie, and an inspiration and a pledge to probably be able to, in the future, to make clinical trials with started in one country, organized in one country in Scandinavia, but enrolling patients across the Nordics. really, really, really inspiring. And I think that is the future for the region if we're going to attract even more clinical trials to benefit the patients and the hospitals in the region. And finally, Lynn, and I think I'll sort of end on that note, showed us a very elegant picture of our cultures in the Nordic in relation to all the other cultures in the world. I'm a Dane, I work daily in Sweden, and there is a lot of differences if you really look for them. But I think what we can agree here is that there is a lot of commonalities. And if we build on those, if we honor the differences, and we start collaborating even more than we do today, then there is a great future, an even greater future for the life science in the region. So with those words, I'll thank you for today. See you again next year. And as I said, there's tapas and wine outside here. So thank you.